Hi, friends, and welcome back to the BioHackers podcast. I'm your host, David James Clark IV, here with the Titan of Trees, Dr. Alex Feltus. Hey,
Thank you,
Alex.
thank you, thank you. That's tied with a capital T, David. Just to make
Yes,
sure
of
that's
course
clear.
it is. Do you see
Yes.
what I did there? Today's topic is trees.
Oh, it is. I didn't. Oh, yes, I did know that.
Yes. Cue the opening song. All right, welcome back. So, as I said, today's topic is trees, and there's a word floating around in my head that a lot of people have been bringing up probably more in the last week than in my entire life combined. And that word is, wait for it, ethnobotany.
Cool.
Whoa. Yeah, that's not a word you hear every day. But I have come to learn that word means the connection, the cultural connection we make between ourselves and trees, or plants in general, but today it's trees. And I got to thinking about the role of trees in my life, and I thought that would be kind of a fun conversation before we bring Dr. Alex Harkess on, who's going to be talking about genomics and sequencing trees. I noticed there's a couple of trees behind you. There's a big palm tree right here behind me.
Yes.
Trees are everywhere, and obviously play a big role in our breathing and other elements of our everyday life. I remember I grew up in a small surfer town in the central coast of California, and we had a ton of eucalyptus trees. And I didn't really appreciate how amazing and medicinal and powerful and tall and oily and smelly eucalyptus trees, are, and how special they are in the tree kingdom, really growing up, because I was surrounded by them and I remember climbing them. They do have that fabulous smell. We had a couple of interesting, pine trees, a few oaks when I was growing up. Then later in life, when I bought my, first or second house, there was a small weed out in the front yard, and I was getting ready to rip it up. And somebody was coming by and said, no, you want to leave that. And I'm like, why? It's just a weed. Just wait. I've been in this house for 19 years, and now that weed is 30 feet tall.
Oh my gosh.
I know, it's phenomenal. It's a gigantic, beautiful pine tree. The trunk is huge, and it protects us. It's right at the corner of our house between us and the street. And I get this sense that, this giant pine is looking over and protecting, my family. So again, trees provide such an interesting connection, between all of us in the plant kingdom. Why don't you share some of your favorite tree stories?
It's interesting because I think of trees and I think of plant more trees, mitigate climate change. And I love to walk in the woods. It takes me a while sometimes after I've been staring at a computer screen to be walking in the woods for sometimes even a couple hours before I feel a sense of relief, like getting back into nature and that kind of connection. But I was thinking about my childhood and sort of instead of this like adult version of trees, right? There was a weeping willow tree in my front yard that I used to climb. It was small enough when I was a little kid to climb up it. My Aunt Lulu had a magnolia tree in Alabama I used to climb all the time. Those were super easy to climb. And just these magical moments, like I can still see myself like inside the magnolia tree, like looking at the house when I was like, I don't even know, 4 years old, like really early memories. And that connection with nature and kind of like my body, I think, with nature, like being able to scale up a tree, you feel so proud of yourself.
Yes,
and
you
just
do.
plain fun when you're a kid,
Yeah.
So there's a huge connection, there's a multiple layers of druidic connections to trees that I don't think about all the time.
Did you just use the word druidic?
I did use the word druidic. Yes, there is a druid component to us all.
That is a fantastic word. Anyway, I digress. One thing, you talk about climbing trees, and I climb trees too, and the idea that it really elevates you, especially as a kid, right? I mean, I lived in a one-story house, And so I had, I didn't have a lot of opportunities to get higher than, 10 feet. But in a tree, you can get up to 20 feet high and see the world from a completely different vantage point.
Yeah.
And that's, another really cool memory of mine is, being up there and like you said, it makes you feel invincible. The closest you can get to flying, I think, as a kid. And the imagination just starts going. Tree houses, one of my...
Oh my gosh, yes. I haven't thought about my tree house for years.
Oh gosh, fantastic. And so today's topic is really meaningful, from that perspective, because we're now going to see the intersection of that, ethnobotanic cultural connection that we have with trees, the druidic connection we have with trees, and science, the science of genomics, and our guest, Dr. Alex your Dr. Alex Harkess, is going to talk about the two Alexes are going to talk about the science of sequencing trees and allowing us to learn from them to build maybe a more intimate connection with trees that are meaningful. And this campus tree project, where if you think about every campus has a unique and special tree that everybody remembers in the same way that we're reminiscing about our childhood. I remember a special tree on the campus of UC Berkeley, growing up. I'm sure Clemson probably has a pretty cool tree.
Right outside my office window, a huge oak tree. Yeah,
exactly. So, how cool would it be to sequence that tree and learn more about it, maybe a little bit more about its history or some of the characteristics, that make it special. So with that, let's welcome Dr. Alex Harkess onto the show. Welcome back, biohackers. We're here with our guest today, Dr. Alex Harkess. How you doing today, Alex?
I'm good. Thanks for having me.
Fantastic. We're really excited to talk to you about trees. Alex and I have a real cultural childhood connection to trees. I know nobody loves trees more than you do. Why don't you tell us a little bit about your story? How did you get into science, and ultimately, what is it about trees that excites you so much?
Well, I thought I was going to be a lawyer, and
great start.
and then I became a botanist, and it was a funny path that led me to that. Something you may not know about me, and actually maybe a lot of people don't know about me. When I was a teenager, I was involved with the Jane Goodall Institute in their youth activism program called Roots and Shoots. And it was this program worldwide, hundreds of groups of middle schoolers and high schoolers who were bound together, for this common mission of how do we change the future in a positive way for all people? And how do we, focus the environment kind of in that conversation. And so when I was a middle schooler and a high schooler, I was one of these kind of youth leaders for this program. And so that led me to some really amazing places, led me to conversations with people like Jane Goodall, that led me to Africa, where I got to see firsthand how people use plants in diverse ways and in diverse cultures. And it fundamentally changed my perspective. I was some kid that grew up in the suburbs, basically, and all of a sudden, my reality shifted in a bunch of ways. Wow. So I thought the future for me was going to be, how do I use law to protect, those kinds of places and those kinds of things. I love the outdoors. I loved green. And I went to Miami University of Ohio for college, and when I was there for, what do you call that when you're signing up for classes? When I was there for...
they do kind of like an admissions fair where you go in and like an orientation, and everybody's kind of telling you all the different clubs you can join. Was there a tree club?
That's the word, orientation. I was there for orientation, and there was no tree club. Actually, Miami was a funny place to be a biologist because it was kind of old school. There was no biology major at Miami when I was there. There was botany, microbiology, and zoology. And I knew I wanted to do something in those kinds of fields. And all of kind of the people who were interested in biology very broadly got into a classroom, and two hilarious professors, David Russell and Jim Hickey, an ornithologist and a botanist, regaled this crowd in front of our parents about what it's like to be a biologist at this university. And afterwards, the deal was you had to come and see one of these two professors, and they checked off on your courses, and that's how they made sure you didn't take 25 hours of credits.
Mm-hmm. Right.
Or something silly like that. And so I get into this line with the botany professor, and my turn comes up, and this professor, Jim Hickey, looks at me and says, "Alex, your courses look fine. What do you think you want to do with your life?" And I remember it so vividly, and I said, "I have no idea. Maybe I'll be a botany major." And it was kind of a joke. And this guy cracks up and says, "You're in trouble." And he says, "Come to me. First day of class, come see me." And so first day of class, I've got my map out of campus. I'm walking around trying to find this building called Upham Hall. His office is in the basement. It's dingy. I can still smell it,
Yeah.
I walk past a room called an herbarium. I had no idea what an herbarium was. And I knock on Jim Hickey's office and open the door, and this guy says, "I've been waiting for you." And I think first day of college, I'm in trouble with a professor. What have I done? I'm 18. Yeah. And then Jim Hickey sits me down for 45 minutes and fundamentally changes the course of my life. He tells me why he studies plants. He was a fern biologist, a taxonomist, and a systematist. He studied how species are related on our planet. And he told me that being a plant biologist is a lot like being a historian, except you're a historian for the history of our planet, and what we study cannot speak to us. It didn't write letters or anything like that. And the way that we tell the story of how our planet has changed over time, over the last billion years, since the first green thing evolved, is by looking and studying and exploring the diversity of plants on our planet, exploring their DNA, 4 letters that can teach us quite a bit, not just about how our planet has changed, but how plants have changed and how humans have interacted with those plants in different ways. And so you can imagine me, an 18-year-old, sitting in front of this professor, just getting my mind blown about what it's like to study plants. Yeah. And so this whole notion I had that the way that I was going to change the world was by studying law and controlling kind of that aspect, I dropped that right away. Yep. And I knew, in my heart that the way forward for me was to tell stories for organisms that can't tell the stories themselves. And that's led me into this really, a career that makes my soul very happy. I'll be a little sappy about that.
Yeah, that's awesome.
So, that's kind of my origin story of why I'm an evolutionary biologist and why I think there's amazing stories hidden within every organism in our planet. As that quote goes, there is a book within each of us.
Yeah, nice. Well said. And I think I love that you're writing the book for those who can't write, but we have a super intimate relationship, obviously, with plants, across the planet, and the diversity that you're talking about, being an evolutionary biologist, diversity plays a key role in that. And there's a cultural connection too, in learning from the plants and the way they interact with the planet and how humans interact with the planet, each other, and plants. So how has that led you to some of the work that you're doing right now and the way that you interact with, your students and your lab and really the amazing research you're doing in your lab?
Yeah, that's a great question. When I think of plants on our planet, I think of it in 2 ways. I think of time, going back to Jim Hickey sitting me down in his office, my first day of college. The first green thing evolved almost 1 billion years ago on our planet. The first plants moved onto land maybe 500 million years ago. The first flower evolved 130 million years ago. And we as a species, humans, have only been farming for 12,000 years or so, the blink of an eye, relative to this amazing history book that has been changing and evolving over time on planet Earth. So I like to keep perspective and remember that in some cosmic way, I am small, I am short-lived. And I find that actually very empowering that humans, despite not being on our planet for very long, can have the potential for huge impact. And I couch that understanding kind of when I train people in my laboratory. There are several 100,000 species of plants alive on our planet today, 350,000 or so, maybe more. It's kind of unclear. And it's a funny thing. I'm at a Genomics Institute. And so imagine if you walked into a library that had 350,000 books, and only 600 or so had ever been checked out.
Yeah.
All the major groupings or categories were there, but we'd only generated genomes or really read 600 of those books in depth. And that's where I see our laboratory contributing to the greater good of plant biology, is to, in some ways, act as a printing press and tell those stories for plants, shrubs, trees, flowers, whatever, that again can't tell those stories for themselves.
Wow, that's cool. And I love the library analogy or the book analogy. I mean, I had no idea that we'd only checked out so few books, and there's a tremendous number of species that still need to be sequenced and annotated. And I don't think we said that you're at the HudsonAlpha Institute in Alabama, and so it's a biotechnology institute. You're doing a lot of interesting work in your lab there. Why don't you talk a little bit about what HudsonAlpha is and how your lab really has helped bring this plant genomics mission to the forefront?
HudsonAlpha is a nonprofit research institute with a mission to improve the human condition using genetics and genomics.
Wow.
It was founded off the backs of the Human Genome Project, right? And, just for a quick history lesson, I mean, that was a 20-year project, billions of dollars, multiple institutions, multiple countries, thousands of scientists, all banding together for, I think, one of the most miraculous achievements of mankind, to produce one human genome that was actually a mosaic of several different individuals, but still one human genome. One of those great multi-country partnerships, kind of, like space travel in some ways, that I think is really brilliant.
And that was the first two books in the library. And that was the human genome and the Arabidopsis genome, right? That's only been 20 years.
That's exactly right. And even the human genome, even though it was published in 2002, it took another 20 years and new advanced sequencing technologies to really finish that one human genome. So almost a 40-year project, basically. And what's exciting about this place is that most of the faculty here are from that Human Genome Project era, particularly from the Stanford Human Genome Center. So this is a pretty excellent place to do large-scale genomics, both for clinical purposes. We sequence, the rural we, sequence genomes for rare diseases and try and match up genotypes to phenotypes. Cancer biology, basic biology about gene transcription. And then, that's how the Institute was founded. How can we use the power of genetics and genomics to improve human health? And then obviously as sequencing technologies became more widespread, plants, took a huge center stage in, driving kind of the importance of not just human health. Obviously plants and animals are intricately tied together in that sense, but again, trying to figure out this library that's largely untouched. Plants in many ways have experienced a lot of the major problems that we're trying to solve on our planet regarding sustainable agriculture, for instance. There are, again, hundreds of thousands of plant species that exist on our planet, some in deserts, some in water, some that are in pampered parts of the country at perfect temperatures, some that are not. And again, there's a time aspect. So plants have seen terrible parts of our planet's history, right? Rocks have come out of space and collided into our planet and destroyed much of the life that existed. For instance, the dinosaurs, except for the lineage that leads to birds today, wiped out. And yet plants have survived and adapted and changed over time. So what I find really brilliant about a Human Genomics Institute at its beginning adopting plant biology is that we get to use the same techniques and technologies in different ways to improve the human condition. And so this is a really exciting institute to be at in that perspective. I walk down the hall and I talk to people working on cancer biology. They come down the hall and talk to me about complex plant genomes. And it's a funny perspective because, again, many people at this institute work on one genome, one species, basically. And then you come over to the plant biology laboratories and we work on hundreds. And although the questions are different, the motivations are the same. How do we make life on our planet better?
That's a really interesting point. And, talking about the connection between the human condition plants, and I really love the historical time element you're bringing in. These plants have lived through climate change, right? Maybe not as dramatic as the climate change we're experiencing now, but there's been other climate changes and they've adapted. And so can you learn from their genomes maybe some important lessons that can help us adapt better to what we're seeing in climate change and other kinds of catastrophes?
Absolutely. Genome sequencing is like time traveling in some ways. So we carry with us scars from the past in some ways, and plants carry those scars from the past in the form of DNA variation or even epigenetic variation sometimes. So again, when you're at an institute like HudsonAlpha, you're basically inside of a printing press. We've got huge capacity of DNA sequencing technology here that, again, is useful for human biology and plant biology. And so what we can do kind of from an evolutionary perspective is start with a question. Are there plants that exist on our planet today that are very good at something? Whether that could be drought tolerance, being submerged underwater, high CO2 tolerance, low CO2 tolerance, the ability to float, anything. And then we can go and find that species, find actually several related species that may or may not do that thing, build a comparison, generate genomic resources, and start to ask in a comparative genetic setting, what is the difference between species A and species B that allows it to do something novel?
Oof, cool.
And then, starting with that kind of evolutionary question, you build genomic resources, you apply functional genomics, and maybe you start knocking genes out and validating that this gene does something useful in this species. And again, it's funny to think about. We might be studying a species that is not a crop, something that most people probably don't care about. But what's beautiful then, you come back to that library concept, and we find a gene that does something very valuable in a species that most people don't care about. Maybe it's a weed or something like that. Well, then you find through evolutionary history, is there a related gene in an important crop species that might do the same thing? And then we tie it kind of full circle in that sense of this idea that the tree of life is, in my perspective, one of the most powerful tools that we have to find new alleles, to find new genes that control important traits that can drive the future of sustainable agriculture.
Very cool.
One of the things I think is so cool about that too is that you can. There's a lot of people that people have genuine concerns about genetically modified organisms, GMOs, and I doing plant genomics a long time ago. I realized that you could find a gene within an organism and then copy that gene and paste it in other varieties, other individuals of that crop, say, and then do a natural crossing essentially by splicing a natural gene, a natural allele into a different variety. And that's just so powerful to do, like inside intraspecific, intraspecies, as opposed to outside of the species type comparative genomics. It's a very powerful approach, very practical, right? Because we need to feed people and grow fuel and things like fuel products and things like that.
I completely agree. I think many of the solutions that plague modern agriculture or the future of modern agriculture, many of the solutions already exist.
Yep.
We're just not looking under the right rocks.
Here. So let's talk about looking at rocks. So, I mean, I'm thinking through a lot of the science that you're talking about, and then the biohackers who are listening, and a lot of the biohackers are computational. And I know Alex is a big proponent of data-intensive computing, and I know you both Alexes are. So, how much of this science or research, this comparative genomics, is digital? All of it? Most of it?
A little bit of both, yeah. I think there's something completely unreplaceable about a good botanist. The ability to explore nature and touch and smell and identify, and just like Barbara McClintock did by noticing the oddities, right? She stared at corn for years right, and found different colored kernels that would segregate oddly in populations, and she could track them over time. And she discovered transposons because of her total focus on identifying, something that was odd. And I think there's kind of, from, again, my perspective and my training, that's where we start. And there's something very, I think, special about being connected to your organisms of interest, the ability to go out in nature and touch them and see them in their natural habitats and identify when things maybe don't look as expected or functioning differently. That's an unreplaceable quality about being a biologist. I think we often have a tendency to forget that plants exist on our planet.
Take them for granted.
It's easy. Yeah, it's easy to get obsessed with the computing, and I'm glad that there are people that do both.
And so, but there is a digital component. So, I mean, you're talking about, you go out, you gather and observe, and then you bring the samples back, the physical samples, you sequence them in these fabulous machines at HudsonAlpha. And then once they've been sequenced, then it's data. And at that point, then it becomes, a computational biology, bioinformatics exercise. So really you need both. And it's interesting,
Absolutely.
In a lot of these podcasts, we've talked about wet lab versus dry lab and, and the idea of in silico, research, which is a cool term, and the power of that. But you're bringing to the forefront, whoa, just a sec, these are living organisms. It's important that we pay attention to, the non-digital elements of it as well. Are there any other kind of projects? I know today the topic of the podcast is training through trees. So what is it about trees that really have captured your imagination recently?
Yeah, so broadly, my laboratory studies how plants reproduce. I'm absolutely fascinated by the diversity of mechanisms that plants have evolved that allow them to reproduce sexually, asexually. Some plant species, maybe 5 to 10%, have separate sexes. And just like me, I'm genetically an XY sex chromosome karyotype. Plants have also evolved sex chromosomes repeatedly, actually, hundreds or maybe even thousands of different times. And so they've, evolved really myriad forms of the ways in which they reproduce. So my laboratory tries to find genes that control sex determination in plants. And again, that's got a very practical purpose. Some plants, like take hops, for example. Hops have separate sexes. Males and females, and only the females are used for that beautiful hop cone that is used to bitter and flavor your beers. You can't do anything with the males other than use them for breeding. And so again, there's huge economic value and practical value in trying to identify how plants determine their sex.
So tell me, Alex, how do you extend your lab into, a more educational, kind of a virtual educational setting? I know we've collaborated on a course at Praxis, and it's kind of the same thing that Dr. Feltus, is trying to do with his Clemson lab, is extend it to hundreds or thousands of people through these virtual classes. How do you approach a class, let's say, in plant genomics?
So I'll back up a little bit. When I was a postdoc at the Donald Danforth Plant Science Center in Blake Meyer's lab, I, with a lot of support from the Danforth Center and with Washington University, was able to develop a couple modules for a graduate first-year class that basically every graduate student in the plant molecular biology program took. And I was really obsessed with this idea of, this was maybe 2017, 2018. Can we democratize genome sequencing? Does it need to be something that only a few select institutes are really good at? And this was right around the time that Oxford Nanopore started really releasing nanopore sequencers that were this size, right? The size of maybe a couple sticks of gum put side by side. So I tried it, and WashU actually paid for this class to buy an Oxford Nanopore sequencer for $1,000, and I developed a 2-week module where we brought duckweeds into this class, and we generated data, on a conference room table. Students loaded this flow cell for a duckweed genome, and we generated a nuclear genome assembly, generated a chloroplast genome assembly. Another postdoc in Rick Vierstra's lab, Fionn McLaughlin, generated mass spec data for the same duckweed. And then it all started falling together that over the course of a semester, students had generated several authentic, real, never done before pieces of data, both genomic and proteomic. And this postdoc and I, Fionn, we looked at each other and said, this is a manuscript. And it's more than that. It's a manuscript with us on it, but also all of the students as co-authors. And many of these students had never written a paper before. They had never done genomics or proteomics. And so this was not just a first entry into the field, but it was something that resulted in a tangible item for them. So we published that paper in the Journal of Experimental Botany, which is, a wonderful journal that I regularly read. And it started to click with me a little bit. I remember asking a student in that class, you're at WashU, an amazing school to be a plant biology PhD student. What do you think was the trick? How did you get here? How did you get to your dream school? And I remember one of these students said, "Oh, that's an easy answer, actually. I had publications as an undergraduate."
There you go.
Mm-hmm.
And it started to click with me that there's inequity in our field, and it's an unavoidable topic in some ways. And many programs are built specifically to address this inequity. So I'm certainly not the first person to ever point it out. I recognize that. But there's definitely some people are forced to pick between, do I work for money on campus or in my college town, or can I work for credit hours in a laboratory for 10 to 20 hours a week? And not everyone is able to make that kind of compromise. And so in some ways, some students are, given the ability and an amazing opportunity to publish as undergraduates, and other people cannot do that. So this all started to roll around in my head that, can we build authentic educational programs that use genome sequencing in classes to create a place where students can publish authentic scientific results, develop hypotheses, generate the data, and publish it. It's something that ends up tangibly on their CVs, that allows them to get that next big grant or fellowship, or get noticed, or build new skills, or meet someone for workforce development that can lead them to their dream job. So I left my postdoc and I started as a faculty member here at HudsonAlpha, and joint with Auburn at the time. And I was tasked with designing a graduate course. It could be whatever I wanted it to be. And again, this was at the beginning of COVID. I started March 1st of 2020.
Welcome to the chaos.
Exactly. Welcome to the chaos. So not only is it COVID, but HudsonAlpha is 4 hours away from Auburn's campus. So it has to be entirely remote. And I was scratching my head thinking, how do you teach an authentic genomics education course from 4 hours away? And that's when Alex Feltus and I first got connected with each other, because of Stephen Ficklin at Washington State, who knew you.
Thank you, Stephen.
So there's kind of the origin story. Yeah, and thank you to Stephen. That's right. So there's the origin story for me. So the next big question is, well, if I'm going to design a class that brings authentic genome sequencing into the classroom, what do you sequence? Duckweed was easy, but I'll be very honest, as much as I love duckweeds, most people don't care about it, and that's fine. About this time, I met a professor at Auburn named Dr. Leslie Gertzen. He's the curator and director of the herbarium on Auburn's campus, a dead plant library, same kind of space that I met a botany professor who changed my life for the first time. I meet a botany professor who changes my life for the second time. Les Gertzen tells me the story of a tree on Auburn's campus from maybe 100 years ago. 2 southern live oaks, Quercus virginiana, called the Tumors oaks, stood guard at the front gates of Auburn University. They are iconic, huge southern live oaks. And after every football game, students would throw toilet paper into those trees. It was the first thing you saw when you left or entered campus. The live oaks were on every logo at Auburn. And they were trees that meant something to that campus. It meant something to the culture, to the identity, to the heritage, whatever you want to call it. And about 10 years ago, those trees were poisoned. with a really toxic herbicide, and eventually those trees died. The university tried to save them, professors tried to save them, but before the trees totally died, and this is what Dr. Gertzen was really telling me about because he was there. Before these trees died, a handful of professors from the horticulture department, the biology department, and a few other places took clonal cuttings of branches, basically suckers, and planted them. Basically, they made clones of these trees before they totally died. And my first question was, do any of these clones still exist? And Dr. Gertzen said, yes, one's in my backyard. And then I knew I had my tree.
My gosh. So that became the guest lecturer for your virtual genomics course, and really the topic of your research.
Exactly, yeah. How do we get people excited about studying plant biology? That's a difficult challenge in many ways. I think the challenges with recruiting and maintaining excellent students, diverse backgrounds, diverse voices is incredibly difficult in the field of plant biology. Not just the inequities, but that exist. Smashing lots of barriers, I think we're up against. People maybe don't have as intimate of a connection to food insecurity and sustainable agriculture as people do to human health. Everybody has a connection in some way to mammalian or human genetics and human biology, disease, cancer, all of the above. But perhaps many people have never stepped foot on a farm before, or thought about where food comes from, or thought about the constantly changing targets that plant breeders, who are some of the most noble people on our planet, are trying to solve. So I started thinking, well, can we show students that the field of plant biology is a diverse, exciting, agile field with huge workforce opportunities? And can a tree be, my door to show students how exciting the field of plant biology and genetics is? So that's what I call training through trees. Can we use them as a
cool
gateway?
Story.
Well, that's a gateway to the skills to be able to assemble a tree genome, or the same skills you need to assemble a mammalian genome too. So you're learning the fundamental tools you need to be able to do genomics in any species, right? When you're learning this stuff, it's pretty cool. I found that the biggest I learned this firsthand when I was doing cancer biology, and then I switched over to plant genomics. And I realized I wanted to help people. That's why I went into this field, and I wanted to cure cancer. But I realized when I was doing plant genomics, and I did a lot of crop genomics and things like that if you really want to have an impact on human health, you do it through food and agriculture. You'll have a much higher impact across the planet than you would if you did biomedical research. They're both very important things, right? But I think it's a very powerful sell that I tell my mostly medical student cohorts to think about it. But people like plants once they realize they're cool, right? People don't realize that plants are very complex things, just like a human is. And some of them are worse than humans, right? And understanding them much worse.
Well, yeah, and Alex, I think you've done a fabulous job over the last 35 minutes or so of making the case for plants, and the fact that they're powerful and cool and important, and and people should get excited about them. So then let's get back to your virtual class. So you brought the tumor. Did your students then sequence it in the class and then run some bioinformatic kind of research on them to publish some novel results about the how that's exciting. I mean, this connection between the skills of sequencing, the skills of comparative genomics, and the skills of publishing all happening in one class virtually in 15 weeks or whatever. That's amazing.
And when you explain it, Alex, could you go back and go back to the graft? Maybe even explain in a little more detail what that is, the graft, and then what you physically do to that piece of wood, and then through the process of assembling the genome.
Sure. Yeah. So these professors who took these clonal, cuttings off of the dying oak trees they rooted them. They put some hormones on the ends of those sticks, basically, and they started to form roots, and then you could plant something. And once you kind of trick a stick into thinking it's a whole tree, it starts to produce leaves, and then it had no idea that it was ever chopped off to begin with. And so I started to put the pieces together in my head that I'm privileged to be at a biotechnology institute with the latest and greatest genome sequencing technologies. Short read sequencing, long read sequencing. Auburn University does not have these kinds of sequencers. Actually, most universities don't have these kinds of sequencers. So what can I do to put this technology that we have here into the hands of any student? So that's when I realized that this oak tree presents a huge opportunity for us to take leaves from it, bring them to Hudson Alpha. We extract DNA, we extract RNA, and we just generated 4 data types. Long read sequencing with PacBio Hi-Fi, short read sequencing, Illumina shotgun, a little bit of dovetail OmniC to scaffold that genome into whole chromosomes, and some RNA sequencing to annotate genes. And it was about $12,000 of cost. And serendipitously, right around that time, there's a yearly giving day at Auburn called Tiger Giving Day. And so Dr. Les Gertzen and I pitched this idea that what if we have students resurrect this oak tree genome by immortalizing it with a genome sequence? And again, a tree that meant something to Auburn that Auburn students are now working on. To me, that's like the ultimate way to kind of think about these ideas that, can we have people who, actually respect that organism in a very different way, be the ones who are leading a project. So, and I started to put together a class that, just as if someone was a PhD student in my laboratory, here's how I teach you how to put together a complex plant genome. Here are the steps, here's the considerations, here's where things go wrong, here's what to look for, and here's how you work on the command line to run these kinds of programs. And what I really try and instill, even in people in my laboratory, is that you will be a better computational biologist if you understand the biology of your organism to begin with. And so combining those two things together, that we're trying to train the next generation of naturalists, of evolutionary biologists, computational biologists, at the same time, people who understand the problems of agriculture, the problems of finding genes that control traits that are going to make the next generation of crops. How do we build education that addresses all of those major problems and has an exit route at the end of it for students to build meaningful careers with those kinds of skills? So I think we often do this, how do I wish I had been trained? And we start with that. And so I started to build a class, in a way that if I were a PhD student today, this is how I'd want to be taught. And so we built a class where maybe it was less of a class and more of a research collaboration, more of a cohort, where there was less of a prescribed path through a semester. And again, plant genomes, as Alex Harkess said, are complex and unpredictable. And I had no idea what this genome would look like, this 750 megabase oak tree genome. So for me, also, students got to see what it's like to make authentic discoveries, overcome challenges in real time when you open up a result and say, that's really confusing. Let's talk it through together. There was no canned lab that we always knew was going to work. And in many ways, I think a semester like that gave students a real insight into how science happens. There's a lot of oops and a lot of mistakes and a lot of things that don't go right and a lot of weird discoveries. And when you show people that this is how the scientific process really works in a meaningful, authentic kind of way, I think it changes people's perspectives on how we move forward as a field and how we train kind of the next generation. So the outcome of this course was 10 students over the course of a semester, and I think these students are incredibly brave. We assembled and annotated and wrote most of a genome release manuscript for this oak tree that no one had ever sequenced a genome for its species before. So we discovered new things. Students took on final projects of whatever interested them. Maybe that was comparative genomics, maybe transposon biology, maybe it was validating the genome assembly. And again, I think what digital teaching platforms like Praxis allowed us to do was to build a space, a sandbox, where students couldn't fail, where they were allowed to explore their interests in the confines of a shared vision, a shared question. And again, this idea that we all started the semester in the same place, and students kind of like, different river streams diverged a little bit. Some people worked together, some people worked alone, but we all converged back together into a single stream by the end of the semester. And I think it's an exciting way to teach because it reduces the boundaries of what students are allowed to do. And I found that when you unleash people's creativity, they typically surprise you.
Maybe the concept of PhD level training will erode, and that kind of training just happens at whatever level it needs to happen at. Like everybody has to wait until they get an undergraduate degree, maybe a master's degree to get to this level of training. But high school students can do it too, if you just teach them how to do it, right, at an early stage. Why should we have education and research be separate things? And you've proven that they can be the same thing. And I think it's mega synergistic when you do that. You've shown it. I saw your students, and they were, just amazing what was happening to them.
I appreciate that. And I like that point about synergy. We did this one tree, and then Les Gertzen said something really funny to me. He said, just about every college campus has a tree, Alex. A tree that means something to that university, means something to the local community or state. And I'll give you a few examples. Hampton University has an oak tree on campus called the Emancipation Oak. That's where Abraham Lincoln gave the Emancipation Proclamation. Washington State University has a tree called the Cosmic Crisp Apple. That is one of the more successful apple varieties that exists on store shelves today, right? Bred by, absolutely delicious. And it's not just trees that have cultural importance to an area. It's trees that have economic value. And Dr. Gertzen was correct. Every college campus has a tree that means something to that place. And so we've started to expand this class to multiple universities. And what I learned from teaching a class and developing content from scratch is that it took 250 hours probably for a pretty good course. Could be way better. And that's a lot to put on a person, a single person doing this by themselves. And so can we not just democratize genomics education for students, can we democratize the way that professors teach courses and the way that we deliver content? There's often no need to reinvent wheels where you don't need to. So now we've been starting to expand this kind of class into multiple universities. And now we're connecting students at multiple universities together on shared Slack channels so that students on different college campuses working on different trees are going through the same kinds of intellectual inquiry and thought and hypothesis generation and problems and struggles and, developing different levels of excitement for different kinds of things. And so my dream is that we create networks of students learning these skills and really building not just their kind of skill set for computational genomics, but building their network, building kind of their cohort of people that they can rely on, people that they will see on the job markets, people that they will see at their next companies, and kind of create this new way to think about synergizing genomics education.
We've been building this biohacker community through these hackathons, and it's been mostly cancer biology focused. But we've talked about doing a plant genomics hackathon, right? If we make that happen, and we'll be able to merge those or link those two worlds, not merge them. Everything needs to be linked and separate, I think, to keep it healthy. But yeah, the exact it's interesting when I talk to you, that's exactly the way I think about things, and we're implementing that too on a different place. And so I wonder how many more people there are like us out there that are trying to make this change happen. Because we always, if you have a question in a class, somebody else has it too. They're just afraid to ask, and that's why you always ask a question. So I think the same thing's probably happening. I think that there is a state transition going on in education, and then we're part of that crystallization that's happening right now. And it's really, I can't wait to see what happens 10, 20 years from now if this stuff takes off, right?
Yeah,
it's
and
fantastic.
I really like the point you just made, that both of you made around connecting research with education, and that they don't need to be separate, because that leads to discovery. And you shouldn't have to go through school so that you gain the right to discover something of value and contribute to society, right? I mean, you can discover and contribute while you're learning. That's the best time to do it. And then we also have that kind of bridge, the BioHacker digital badge, that bridge to workforce development. And you badged each of your students from a genomics perspective as well, so that they could then take some sort of validated credential, into the job market if they're interested in doing that. But just coming back to your comment about networking the students and different universities, but also networking the professors and the content developers. Typically, they're the same person, like yourself. Both of you are both content developers and professors. But if you have some resources or some lessons or some labs in your class, Alex Harkess, that somebody at another university could use, no reason to reinvent the wheel, right? So that other professor can then take that content and, make it their own. Maybe it's a different tree, but they don't have to rebuild the course from scratch. Is that kind of the approach?
Yeah, I think that's right. And people do this already. You see professors sharing their lecture slides on GitHub for diverse courses and their laboratories. And it's something really beautiful about kind of the genomics community in general, is that people care deeply about other people, I feel like, at heart. And everybody wants to make life easier for somebody else.
Yep.
I think that's one of the best parts of being an academic. So certainly, yeah, this allows us to do that in that same kind of sense. And certainly I've used content from other people. I've developed other content for other kinds of groups.
So when you talk about the unit of a class, and we've been talking about this class that you taught virtually through Praxis AI to Auburn students, but that concept of a class is starting to morph and change, and also the location of a class. I know now you're at HudsonAlpha, and HudsonAlpha has an outreach program, and you're starting to think about the class as something that's more community oriented. Maybe, you bring it to other colleges, or you bring it to high schools. Why don't you describe a little bit, about how you're connecting with the education outreach program at HudsonAlpha and kind of redefining the concept of a class? So the education outreach program at HudsonAlpha is unbelievable. There's 20 full-time staff from pedagogy development, educators who run summer courses, short courses, lifelong learners, and beyond. And I guess this is kind of a candid podcast. Good. I'll tell you a wacky idea that I've been thinking about for a while. I give talks at the education outreach group to so many talented middle schoolers and high schoolers. And a lot of those people come up to me and, very nicely ask, can I join your laboratory? And this is a problem with every laboratory in the world, is that I only have so much room. And so I've started to try and think outside the box of, does someone need to be in my physical laboratory space for them to contribute to the goals of my group very broadly? And I think teaching a course, or several courses now like this, has changed my perspective on that. And there is data abound in this world. For instance, with plants, there are thousands of Illumina short read data sets that exist for hundreds, thousands of plant species on publicly available
sites like SRA. On the other hand, most people do very little with that kind of data. It's used for a very specific question, maybe to look at mutations in a single gene, and we sequence the whole genome to do that. And 99.9% of the data was never even looked at or touched. And that's fine. That's the way science works in a lot of ways. But what if we could use all of that data for those diverse plant species for something, again, coming back to my motivations, what got me into botany at the very beginning? Can we use that data to build the tree of life or to improve the tree of life in some ways? Can we use that Illumina sequencing data to build chloroplast genome assemblies, which are pretty easy, tiny 150 kb circles? And could people like high schoolers drive those kinds of initiatives? What if we had a list of every publicly available data set online and a list of every chloroplast genome that existed for a plant, and high schoolers knock them off one at a time and contribute to a real body of authentic scientific discovery, something that is no fluff, important, and straightforward to do in a way that is scalable, right? A way that I don't have to cram my laboratory with new chairs and desks, but something that we can still guide and lead kind of, from a computing standpoint.
Again, what's the difference between research and education? At the high school level you're talking about there, right? What if you have a whole bunch of people who are getting papers for their chloroplast papers at high school, and maybe they're coming from backgrounds or places with low resource environments, and then they have papers, and they all of a sudden they went from maybe getting into a state school to getting to, an upper tier school or wherever they want to go. I mean, it just opens up so many doors. That is such an exciting, that is not a kooky idea. That is a profoundly awesome.
Yes. It sounds a lot like a perpetual hackathon and something we should definitely collaborate on. I really like the idea of, leveraging the Praxis platform. We do some hackathons in Discord. I mean, there's other, Slack you talked about, other services that we could use to bring everybody together and, have them connect on a weekly basis, have them collaborate with each other, teach them the skills they need, badge them, around those skills to do the chloroplast assembly and the annotation and all of that. And then, start and publishing. I know Alex Harkess, you also in one of your classes, you teach your last learning path is about publishing and how do you publish on GitHub? How do you publish, in journals, peer reviewed and otherwise? I mean, so just a really awesome creative idea and a great way to get those 100 high schoolers and middle schoolers who want to be in your lab, Alex Harkess, get them in your lab, but it becomes a virtual version of your lab.
I think COVID changed a lot for a lot of people. And what I've really appreciated is that no longer can I rely on excellent people coming to me. It certainly happens, but what can I do to bring content to people? What can I do to reduce barriers for students to enter and stay in our field? And the more you put something in front of people, the easier it is for them to take it. And so I've really appreciated that not everyone can be in Huntsville, Alabama. Not everyone can move here. Not everyone can be in this kind of space. So what can we do to democratize kind of the privilege and the resources that we have at a place like this?
Yeah, no, we had a wonderful podcast with Dr. Taisha Farmer, somebody that you know, and she told a story of growing up in rural Alabama, and really didn't even know about these scientific opportunities, didn't have access to the education or any of the resources you're talking about, but somebody reached out to her and really changed her life in much the same way that the botanist, changed your life, and brought her into the fold. She's now a professor at AAMU and HBCU. She's working with you on a couple of grants at HudsonAlpha. So a wonderful example of, an outreach that made a difference, in a huge difference in her life. And now she's dedicated to doing the same for others. So, again, I think it's a beautiful dream, and it's starting to come true for a lot of people. And the digital platforms that we are describing today make it allow us to scale it even further. So, so one of the questions we always like to ask our guests is, what is a biohacker to you?
Is opportunity where it previously did not exist. I think this gives us the ability to, much like Dr. Taisha Farmer, to create doors for people where walls existed and to hold those doors open for other people. So for me, that's the most challenging thing about being a scientist, is to, how do we identify people that need opportunity, and then how do we create it for them and put it in front of them in a way that allows them to drive change.
Fantastic.
And
Thank you.
for
Good
me,
answer. I'm
that's
putting
what
that on the
BioHacker can be.
website.
Awesome.
So the reason we asked that question is, again, you brought up the idea of opportunity. We've always been convinced that the change agents, the people that are going to make the most positive impact to the planet Earth and the people living on the planet Earth, are the people that currently don't necessarily have access to the resources or skills that they need in order to affect that change. So we're just, again, the concept of democratized access to skills and resources, we're trying our darndest to, give everybody the opportunity to hack the bio in a positive way, whether that bio is plant or animal or human or, whatever, and whether that the problem is, climate change, disease, agriculture, food, whatever it is. So great answer. Opportunity. That was a good answer. So as we start to wind down the podcast, I always love to offer up an opportunity for closing, ideas, closing questions, closing comments. So I'll start with you, Dr. Alex Harkess, the guest of honor. Is there anything on your mind, any kind of closing points you'd like to make for our biohackers out there?
I like to tie things back to evolution, I guess. And I think what I find special about plants, trees, whatever, also people, communities, civilizations in general, it's not that different than an evolutionary concept. What is evolution? It's change over time. And in order for there to be change over time, you need diversity. And so I think that is maybe the most important thing that we can move forward kind of in the front of our minds of how do we increase the diversity of the people who are able to access these kinds of resources, and on the back end, the diversity of career options that are now open to those people.
I'm going to let you follow that one up, Dr. Alex Feltis, because that was a good one. Okay, perfect.
I'm gonna go a little bit weird with it. So there's a movie, I think it's from the 1980s, called Brazil, and I believe Terry Gilliam made this movie, and it's a really weird movie. If you haven't seen it, it's a great movie. It's just super weird. But it's like a dystopian future. This is kind of the way I remember. I haven't seen it in years, and there's bureaucracy has gone amok, and the society is just a mess. You can't get anything done. It's just an absolute disaster where humans have gone. And there was this person trying to get their air conditioner fixed, because they were calling all the bureaucracy, trying to get the channels to get their air conditioner fixed. And then all of a sudden, a guy comes into the apartment and fixes the air conditioner. He comes in, like, swings in on a rope, fixes the air conditioner, and the person's like, "Why did you do that?" And he's like, "We're all in this together." And then he leaves. And then I think he shows up a couple more times in the movie, right? But it's this weird thing, like, what in the biohacker in the world is it? Why is he doing it? But then, I what I was thinking about when Dr. Harkess, who I'm a huge fanboy, and I got to get your autograph when we actually see each other, that it's almost like you feel like when you're trying to do things and you have this idea in a university or whatever, any walks of life, trying to get things done, and you're doing that, and then you're swinging across the city fixing air conditioners, then you run into somebody else doing the same thing, and then you look over in the distance, and after a while you start to see more people kind of just going outside of the system and just doing what's right and fixing it. And that's, Dr. Harkess, that's what I see you as a person who's just incredibly, you said, I forget where you used the word, kooky idea or something like that. But I think that you're right there in the middle of, you've got all the fixes that we need, and you're not keeping them to yourself in a lab. you're scaling and replicating those outside of your lab. And it doesn't, I mean, I don't even know if, like, you know, the atmosphere of the earth is enough to hold in your ideas, right? If maybe these will propagate out into the solar system, right, and keep on going. But that's what I see with this, is just like, you know, we're all in this together, and we've got, I mean, I hate, it's so negative and nobody wants to hear it, but we've got dystopian existential problems that are looming for our species, right? And we've got to attack them, and people that are looking at this as an opportunity, you know, for us to evolve and into something better as a species. I feel like people like you are the catalyst for that, and I thank you for all that you do. And I look forward to continue to learn from you on how to do it better from my end too, and teach others, teach our students, right, how to do this. And then it becomes, it gets taken for granted that we live in a, you know, utopian society where discovery is the driver, and, you know, fact-based decisions are the drivers of our society.
Wow. Great answer. I mean, well played, Dr. Alex Harkess. Okay, so I am opting out because I cannot top those two. You guys are the winners of today's game. So thank you again. I think the real winners are the biohackers who are listening. And thank you again to our guest, Dr. Alex Harkess. Fabulous, fabulous episode. Thank you, co-host Dr. Alex Harkess. And until next time.